Such a pleasure to see everybody. My name is Kate O'Brien. I'm the director of the Department of Immunizations, Vaccines, and Biologicals at WHO. I'm thrilled to be here with all of you and to be with my colleague jointly opening this session. So please go ahead.
So it's Manolis, and I'm working as a PID physician in Greece, and currently I'm the ESPID president. So welcome everybody to this first plenary session of the meeting. Thank you.
And we've got such a great room full of people here and great speakers. This session is the plenary session. It's the opening plenary session on pandemic preparedness. And I need not say many opening words, except to really want to acknowledge that on Friday, after 3 long years, WHO did announce the ending of at least the declaration of the pandemic, the public health emergency of international concern, ending the acute emergency phase of this pandemic. And there's so much to learn, so much that we've all been through, so much that communities and countries have been through. And so this is what we're going to talk about in this plenary session, and it's my honor to introduce the first speaker, Maria van Kerkhove, who's been the technical lead of the COVID-19 pandemic response at WHO. And I'll turn it over to Maria to take us up to the podium and take us through the first presentation. Welcome, Maria. Thank you so much.
Thank you, Kate. Good afternoon, everyone. I actually can't see many of you, but I'm really happy to be here in person and see that you are in the room with us. So a little provocative, I actually changed my title to "What Did We Learn?" to "What Are We Learning?" So as Kate said, the Director General of the World Health Organization declared the emergency of COVID-19 over, but COVID itself is not over, and unfortunately, we are still in a pandemic. So I will start by when the Director General announced a public health emergency of international concern. That was on the 30th of January 2020, and on Friday last week, this is a picture from the press conference, and Kate was up there with us, and I wish I had a picture across all of the speakers up there. I've had the pleasure to be doing press conferences with Kate throughout this. The Director General announced on advice from the Emergency Committee that the emergency phase, the crisis of COVID-19, is over, but COVID still exists, the pandemic still exists, and there's much more work to be done. So I have to start there. This is just the latest epi curve that you've seen, and while we are in a much better situation than we were 3 years ago, there are still thousands of people dying every week. There are hundreds of thousands of people estimated to be in hospital with COVID-19, and there are millions of infections each week. This virus is circulating in every country on the planet. We are managing it much better. Population level immunity is much higher, but the threat is still there. And so there are many lessons. There's been hundreds of reviews that have been conducted for COVID-19 and previous epidemics and outbreaks and pandemics. And we need to learn these lessons in real time. This isn't something that happens at the end and we start over. We are learning these and we course correct as we go along. So there's a number of these on the left-hand side, on the right-hand side, excuse me, of the screen. I'm only going to cover a couple today because I could spend the next 4 days going through lessons learned, and I won't. I will keep to my time. But I'm going to focus on a few key lessons that we continually learn and continually forget. The first is about acting fast. The second is around science and the foundation in which we operate. Trust and leadership. You can have the best plans in place, but without trust, without leaderships, your plans will fail. And I argue that pandemic preparedness is now. It does not start and stop. It is a constant. Responding to COVID-19 is pandemic preparedness for the future. So let's start with the first one, acting fast. As we know, we are learning from this devastating effect of COVID-19. Every single person on the planet has been affected by this invisible pathogen, whether you have lost a loved one, whether your future has been altered. Every single person, every single family, every single community has been impacted by COVID-19. But of course, this isn't the first outbreak that we have dealt with. It may be the first outbreak you have dealt with, maybe not in this room, but certainly on the planet. Many people are not experienced in dealing with infectious disease outbreaks. This pandemic has had 8 million estimated, 8 million, around 7 million confirmed deaths, and that estimate is probably 3 times higher. There has been a loss of $16 trillion in estimated revenue losses in international sectors. We've had more than 1.6 billion children out of school. The impacts are way beyond health. And there are more than 300 million people in need of humanitarian assistance as a result of climate disasters and geopolitical crises. And this will happen again. So countries that did well in the beginning, that fared better in the beginning, are countries that had experience. Experience with SARS, with MERS, with avian influenza, with Ebola, with Marburg, with Zika. And these are just some examples. There was a trauma, there was a muscle memory, and there were actions that were taken to be better prepared. I spent some time in Southeast Asia doing my PhD in Cambodia, and in particular, and Cambodia knew how to use village animal health workers to detect something different in that poultry flock, to report that instance of a change in mortality, kick it up the chain so that actions could be done. We saw across the Mekong, we saw across Asia with dealings of SARS, with avian influenza to know what to do, and they took the threat seriously immediately. Whereas other countries around the world, there was this notion that the threat is over there, it's not here. We have health systems in place, we can handle it. We can handle the onslaught of patients. And we saw country after country really be overwhelmed extremely quickly with COVID-19. So I want to bring you back to the beginning. And one of the things that we did on the 31st of January with the first instance of this ProMed article in this red letter banner, we immediately at WHO activated what we call our incident management structure, which means we bring together people across the 3 levels of the organization. This happened within 24 hours of learning of the cluster of pneumonia of unknown etiology. This is an early EpiCurve, and within that, on the 1st of January, we activated that incident management team, and we immediately brought together the questions and asked for more information from China about the cases. We issued our first alert to all of our member states on the 5th of January, which outlined the cluster, what we knew, what needed to be done, reporting information. Within 2 weeks, we issued the first set of comprehensive guidance around surveillance, laboratory detection, infection prevention and control, clinical management for dealing with severe acute respiratory infections. We issued the first protocol with our partners, our lab partners around the world, so that PCR test kits could begin to be produced. And on the 30th of January, the Director General declared a public health emergency of international concern. And when this happened, there were less than 100 cases outside of China. Within a few days after that, we issued the first global plan. I'm going to skip the rest of this video. That's the first thing. We were in a position to be able to act fast, and many countries acted fast. The second thing I wanted to mention is around science and the foundation. We're at a scientific conference, and all of you study science, study public health, are in various different fields. And we know how critical the information that is gathered early in an outbreak. And in the beginning of an outbreak, we want to know, what is it? How is it spread? How far has it spread? What disease does it cause? And how can we stop it? Those are the key 5 questions in the beginning that we ask at every outbreak, every epidemic, every pandemic. There may be variations of those 5 questions, but those are the key elements. And the data that exists in the beginning is very, very limited. Science is one of the major aspects of being able to generate guidance, evidence, advice, but it's not the only essential element. My role within the pandemic was as our health operations and technical lead was to work with the world's expertise and all of our expert networks across many different technical disciplines to translate evidence into knowledge and to translate that knowledge into action. And science underpins all of this. It drives our understanding. And of course, as you know in this room, science changes, science evolves. And as such, your guidance needs to evolve, needs to change. And we turn that into guidance, evidence-based guidance. And then that guidance is turned into policy, into interventions, into action on the ground. Now, this is something obviously that is incredibly difficult to do in the beginning. What we did in the beginning is we used our MERS coronavirus guidance and the package of materials that we had, whether it was guidance, whether it was training materials, whether it was questionnaires, whether it was toolkits to basically ask questions, how ready are you? And we adapted it very quickly for a novel coronavirus. Remember in the beginning, we didn't know it was SARS-CoV-2. We didn't know, it wasn't called COVID-19 at the time, we called it nCoV. But of course, decisions and policies are informed by the evidence, but they have to take into consideration many other factors. So guidance is not the same as policy. So what we do at WHO is we develop this evidence-based guidance through an entire pathway, iterative pathway of data generation, data synthesis. Not all data is the same, of the same quality. Turning that knowledge into guidance, into recommendations, issuing that guidance. And then it's delivered in the field by our regional offices, our country offices, and the ministries of health and the people that we serve in country. And then there's a feedback loop. As more data gets generated, all of this guidance material gets updated. But the policies that are set in country is not the same as the guidance. Think of mask wearing, for example. Do masks work? Where are they most effective? Which population should be using them, and where and why? And how those are used in countries are the policies that are set by the governments themselves. And you have to take into consideration many other factors: context, communities, public opinion, equity, feasibility of their implementation, affordability, acceptability, and sustainability. And science is critically needed in public health emergencies. And in the beginning, of course, it's very difficult to have enough information to make those recommendations. And it helps inform the policies that can lead to more effective, efficient, and equitable interventions. And of course, in every outbreak, in every public health emergency, initially not much is known. And there's limited tools that are available. Remember, during the first year of this pandemic, we did not have vaccines, we did not have therapeutics, and we were using public health and social measures to minimize the impact, minimize transmission as best we could to reduce the pressure on the healthcare systems. And our healthcare workers were delivering the best clinical care that they could to save patients' lives. And we need clear science-informed policies which can be tailored. There is no one size fits all. They could be tailored and useful to the populations that need to use those interventions. Let's move to trust and leadership. So without trust, without leadership, and I could probably spend a couple of hours on each one of those, even the best plans will fail. We have seen over and over across countries, across continents, where we had leadership that failed their populations, plans failed. When communities did not trust the information that was coming, the amount of misinformation and disinformation that has circulated during this pandemic, it's still rampant to this day. The best plans in place will fail. And so the factors that are challenging, what you'll see on this with this GIF here is something I pulled off Twitter actually this morning, which I think is quite interesting. And it's a look at papers that have been published over the last 70 years. I have not verified this information, I should say, but it's just demonstrating some of the topics of papers that have been published over the last 70 years. And what you will see in 2020 is the introduction of COVID-19. That amount of information that came at us in the beginning of this pandemic was truly overwhelming. We had arranged with all of the major journals to share publications that were submitted for publication, not preprints, not publications that had undergone review, but the actual submitted manuscripts to be shared with me and with another individual who was part of our science division. And I read every single one of those papers in the first few weeks of the pandemic. I stopped very quickly because it was absolutely overwhelming. And I think all of you know, any of you who have reviewed for journals, you know that some of the papers that are submitted are not of the best quality, let's just say. And the peer review process has really, I have a deeper respect for the peer review process, I have to say, after the last 3.5 years. But in the beginning of a pandemic, you are pulling together every shred of information that you can to make the best decisions that you can make. And with poor information or with papers that have been retracted, that erodes trust in science. And I think the attacks that we have all seen and we have all witnessed, I hope none of you have dealt with that personally, have truly been substantial against science, against scientists, against public health professionals. And that is certainly something I was not prepared for during this pandemic. But we also have to recognize that science is only one factor. Major emergencies are not led at a country level by the health sector. The health sector is incredibly important, but all of these responses are multi-sectoral. They need to be an all of government, all of society approach. So the health sector may not be the one that's in charge. And decision making is heavily influenced by many other factors, including political factors, economic factors, other interests. And of course, COVID, when COVID was introduced, was not the only crisis that countries were facing. Most of the world stopped everything else and focused on COVID. And now what we're trying to do is deal with COVID as a pandemic, maybe not in the emergency anymore, but dealing with COVID in the context of everything else. It's very difficult to communicate with some decision makers about what the science is telling you. You can say, but the science says this, that means you should do X, Y, and Z. But you have to take into consideration all of the other factors when those policies are set. And again, not all science is good science. Even in some of the top journals that are out there, I think all of you have seen some pretty rubbish papers. And it's confusing because on Twitter every morning I would wake up and I would see another paper, I would see something that would be announced, which at one point was incredibly helpful, but it's too much information. This infodemic of information is very difficult to synthesize. And doing those press conferences every day was certainly a challenge that we faced to provide answers, to be able to synthesize what we knew, what we didn't know, and most importantly, as the World Health Organization, what we were doing to find out with our partners around the world. So there's the difference between evidence-based policies and policy-based evidence. And some countries needed to change their policies to get people back to work, to say we couldn't have an isolation policy for this long because we had to get people back to work. And that's a conscious decision that has to be made, but we need to be transparent about why that is happening. So just lastly on this point, that trust is hard earned, but it's easily lost. And I think all of you in that room know, because outbreaks begin and end in communities. You are part of a community. They don't begin somewhere else and affect this other population. These are the people that we share a planet with. And these viruses and these pathogens do not respect borders. And countries are in a very different situation. While we're in a global pandemic, every country is facing a different situation in terms of how they are handling it. There is no one size fits all. Regardless of all of the different elements that countries are using, how they apply them are slightly different. And it's dependent on what strategies they followed in the beginning. Did they follow a containment or a suppression strategy in the beginning? Or did they follow a mitigation? There was this false dichotomy in the beginning of either you did containment or you did mitigation, when in fact the global strategy was elements of both. And so there are a lot of different factors. And one of these factors is public trust, societal engagement, and unrest. And not only are we dealing with health crises, we're dealing with the COVID crises, but other health crises. So many children miss their vaccinations. So many people miss their cancer treatments, miss their preventative care. And we are now starting to see excess mortality for other diseases. And this really is such a tragedy. In many countries, and I'm sure all of you and your families are facing this directly. We also have non-health crises. We have floods, we have droughts, we have war. We have displacement, we have economic crises, we have challenges that governments are facing, and the fiscal space and the space in which we can deal with COVID is shrinking. We have to make room for everything else. So the argument is pandemic preparedness is now. We can't finish, quote unquote, finish dealing with COVID. This virus is here with us to stay. We are managing it, we are living with it, but we need to do even better. And we cannot lose the gains that we have made in the last 3.5 years for COVID, because if we miss this opportunity, we will again start over in many respects for the next one. But because SARS-CoV-2 is still circulating with us, this virus is changing. There are 1,000 sublineages of Omicron that have been in circulation. Most of these are of the XBB.1 sublineages right now, but there is no one particular sublineage that is dominant everywhere. It's evolving, it is changing. This virus is circulating unchecked. With population level immunity, we are not seeing the level of impact that we saw before, but with this virus circulating, there's more opportunities for it to change, to recombine, to establish new reservoirs in animals, spill back, spill forward. We have to be very careful, we have to be vigilant, and we need good surveillance. So we have to keep up our surveillance to track the known variants and detect new ones. All of this information not only is important for national governments to take stock of what's happening within their own borders, but for global entities like WHO with our technical advisory group for virus evolution to monitor and to assess each of these in terms of their phenotypic characteristics, when we are particularly looking for variants that have a different disease severity profile. So far, Omicron sublineages are similar in terms of their severity, but our worry in a worst case scenario, we would have a more transmissible, more severe virus that would start circulating, and our interventions such as vaccination would not be as effective. We just have to be vigilant. So what we're trying to do with countries is to calibrate the response. The worst thing that could be done, that the Director General said on Friday, was to dismantle systems. We need to calibrate the response. We can't keep it up at the level that it was at the height of the pandemic, but we can't dismantle them either. So we want to maintain the investments that are there. The laboratory-based systems. There are countries across the world, Somalia, Afghanistan, had no PCR capacity before this pandemic, and now have PCR testing capacity across the country, have the ability to do genomic sequencing. That is not just beneficial for SARS-CoV-2, it's beneficial for any infectious pathogen that is circulating. These are investments that need to be maintained that will benefit the future. So we've issued a number of different policy guidance to help countries calibrate what needs to be maintained, what could be let go, so that we adjust as we go forward, keeping that workforce, keeping that knowledge base, and using the last 3.5 years to start as the foundation going forward and not backtrack. And these are the planning scenarios that we outlined in 2022 for 2023, and these are applicable for this year and into next. We have a base case scenario, which is largely what we are seeing right now, where we see the virus continuing to evolve. The severity, the impact is reduced because we have better clinical care. We have patients entering the clinical care pathway early. We have antivirals, although access to antivirals remains challenging around the world. We have population level immunity that is very high across the world. But in the 4th year of this pandemic, we see waning immunity, of course, which is why the SAGE recommendations are focusing on primary series for those who are most at risk, people over the age of 60, people with underlying conditions, our frontline workers, targeting those populations for 100% coverage of primary series and a booster as recommended by the policies set in country. We have a worst case scenario, which we're planning for, which governments need to plan for, a more virulent or highly transmissible variant, which evolves, which emerges, and which our vaccines are rendered less effective, where we have immunity against severe disease and death wanes rapidly. Again, we have to prepare for this. We have to plan for this. We have a best case scenario as well, where future variants are less severe. This is this theoretical where SARS-CoV-2 quote unquote becomes the common cold. That narrative that Omicron was mild was a deadly narrative. In many countries, the amount of death, not necessarily the death rates, but the amount of death in some countries was higher during the Omicron wave than previous waves because of the sheer increased transmissibility of this particular variant. And so we have issued an updated global plan. This was issued last Wednesday before the Public Health Emergency Committee met, and we did this on purpose because we wanted to outline a 2-year strategy to continue to support countries who've already started this in transitioning from a crisis response to longer-term sustained COVID-19 response in the context of other health challenges. We still have similar goals: control the incidence of SARS-CoV-2 infections, particularly in high-impact individuals, and to prevent, diagnose, and treat COVID-19 to reduce mortality, morbidity, and long-term sequelae. And here we're talking about the impacts of repeat infections, and we're talking about post-COVID-19 condition. And I can give you an entire talk on post-COVID-19 condition, a burden that we are going to have to deal with for the next many years. And of course, as WHO, we're supporting member states to transition from crisis to longer-term integrated respiratory disease management with influenza, but also making sure that we think for the next year, 2 years, 5 years, 10 years, and beyond. So everything that we are doing is preparing for the next one, strengthening global research capacities for future pandemics, enhancing surveillance, testing capacity, sequencing, and public health intelligence, the analysis that we use, the data that is generated for a purpose. Equitable access to medical countermeasures. So there are many therapeutics that are available, but the access to these therapeutics and antivirals is not equitable around the world. Part of this is driven by cost, and there are some generics that will be coming online and that will help increase demand. There is a huge amount of work that has been undertaken to increase medical oxygen access and sustainability around the world, and that needs to be sustained because it's not only good for COVID, but it's good for pneumonia, it's good for other diseases. We have to continue to work to develop trust by engaging communities, empowering communities. There are many surveys that are out there that have shown that trust has plummeted around the world. And this is something as scientists, as public health professionals, as humans, we need to work on because there's a huge amount of misinformation and disinformation that is circulating. We need to build and scale resilient healthcare systems. Our healthcare systems are incredibly fragile in high-income, middle-income, low-income countries around the world. They have taken a massive beating. Our healthcare workers around the world have done tremendous work in the last 3.5 years, and they are exhausted. They are overworked, they are underpaid, the underlying health infrastructure system has been weakened, and it needs to be reinforced. And we need to invest in this long-term pandemic preparedness, this cycle of panic and neglect. We were in a cycle of panic and neglect throughout the entire 3.5 years. You remember every new variant that came, it made the headlines again. But this virus just circulates and circulates, and it takes advantage. There is no seasonality yet. There is no predictability yet. It is taking advantage wherever it can. So there are longer term plans. So the DG outlined 10 proposals to build a safer world based on more than 300 recommendations and this new global health architecture, strengthening the health architecture for health emergency preparedness, response, and resilience, focusing on governance, systems, and financing. Across the board, we need to do better. We collectively, every single one of us, every single institution, every single organization, every single politician, we need to do better. We have to remember these outbreaks, these pandemics last a lot longer than a political cycle. So it needs to transcend us. It needs to transcend our lives, our children's lives, and our grandchildren's lives. And the reason that this is so important is because it's going to happen again. So in the last 30 years, we've had multiple outbreaks. This is actually just the last, only up to 2020. I have to update this in recent years with Marburg, where we've seen Marburg in countries that we haven't seen, that had never detected a case. We will continue to see the emergence and re-emergence of known pathogens and new pathogens. COVID-19 was disease X, and the next disease X is out there. And the reason this is so important is because these viruses don't respect borders. And 70 to 80% of these are zoonotic. The way that we live on this planet is changing the frequency, are changing and increasing the drivers of spillover and amplification. Climate change is enhancing this because, for example, mosquitoes, where mosquitoes can transmit, that's changing. The geographic range is changing. And we have the known threats, the Ebola, we have Marburg. MERS coronavirus has not gone away. Detection has gone down because surveillance has gone down, but it has not gone away. H5N1 has expanded massively. Who remembers Zika and microcephaly from Zika? And again, anywhere these pathogens emerge, they can be on the other side of the planet within 24 to 48 hours because we live in an interconnected world, and our ability to do detection, although surveillance has increased, is being challenged because one could argue that with all of the things that have happened in the 3.5 years, in terms of the geopolitics at a global level, nobody wants to find the next pandemic pathogen. We need researchers, public health professionals to study in markets, to study in the field across all of the different types of animal, wild animal, domestic animals, along the animal trade, making sure that these pathogens are studied under biosafety and biosecurity rigor. Because we need good surveillance, we need good transparency, we need good collaboration for earlier detection and earlier action. And this is a map that's looking at, this is a project that we have, and it's actually just draft because we haven't finished it yet. But what we are looking at is mapping over, remapping the spillover of pathogens with epidemic and pandemic potential. Now we're building on maps that exist. I'm sure you've all seen Ebola maps and Marburg maps, H5N1 maps, independently, separately. What we're looking at is the composite risk, looking at the different covariates in terms of environmental factors, animal movement, population movement, density, and looking at the aggregate risk. Within this map, we have chikungunya, dengue, Zika, Hendra, Nipah, MERS, Mpox, plague, Ebola, Marburg, Lassa, SARS-CoV-2, and CCHF. We don't yet have H5N1, which is this map here. The expansion of H5N1 in the last few years is dramatic across many different countries, spillover into mammals, into humans, and flu is a certainty. We have seasonal flu. We will have another pandemic of flu. When is the big question, and we need to be prepared. And the reason this mapping is so critical is because what we want to do is we want to do better at the local source so that we have better detection where spillover is happening as best we can to mitigate that spillover from becoming an outbreak, becoming an epidemic, becoming a pandemic. If we do better at this mapping, we can have better risk communication. We could have better lab algorithms and making sure that the right sample is sent to the right lab with using the right detection methods across the board. So we need to do better. And I will end by saying that the world pandemic preparedness is a constant. What we want to do is move towards the left of this slide here. We want to be able to prevent animal to human spillover and human to human amplification, increase preparedness and capacities, and have operational readiness for the known pathogens and the pathogens that will come from those viral families or the bacteria that we think might cause that next outbreak. We want to be able to contain as much as possible, to mitigate as much as possible, so that these spillover events do not become the next pandemic. And I think with that, I will stop. Thank you very much.
We thank you, Maria, for keeping the time and for this impressive presentation, actually. Yeah. Our both next speakers will join us remotely via Zoom. I think Scott is with us already, and Scott Dowell, and then it's the Colin Russell. They're international speakers. Scott is currently based in the USA, and Colin is currently based in the Netherlands. So Scott, are you here? Yeah, you are here. Yeah, it's okay. Yeah, I can see you. Scott will spend the next 10 minutes with us and mixing art, filmography, and science. He will report us on a quiet news from the Western Front. So Scott, please go on.
And as Scott's just getting ready, perhaps I can also just remind folks that for your questions, we will have a question and answer session. And for those of you who are online, your questions can be submitted by the question and answer virtual platform. For those of you in the room, we have microphones, but you can also use your Ask the Speaker function on the app using the interactive feature. And we really welcome those questions. We're not seeing any coming in yet virtually, so please submit them online and in the room. Thanks so much.
Good morning, greetings from Seattle, it's still early here. We're on the far western edge of the planet. And I have to say first, I'm really disappointed that I couldn't be there in person. But hi to Maria and Kate and all the friends there. Adam had asked me if I could give a short bridging talk between Maria's review of the pandemic and Colin's discussion about H5N1 and the future. And so I said I'd be happy to talk about our view from out here on the Western Front in the early pandemic. What I remember in the first days and weeks of January were watching the news and data coming out of Wuhan, and Keith Klugman walking into my office and saying, these early reports are really concerning. The epidemiology is worrisome, the clinical features are worrisome. And then we started seeing reports of cases outside of Wuhan, case in Thailand, Japan, and we sat there with this literally an envelope and wrote some numbers on the back of it, trying to figure out what numbers of cases had to have been exported to detect the cases that were being detected. And we concluded at that point that there had to have been thousands of cases exported. It wasn't long after that the Seattle flu study, which we were involved with here in Seattle, was detecting the first cases here. And it was confusing at first. We had one early case and then a much later case that were confirmed, and the epidemiology connecting those cases only became clear over time, but it did seem likely that we had prolonged and a more considerable transmission outside of Wuhan than was being appreciated at the time. And then there was the Diamond Princess. Remember the cruise ship that was stuck in Japan for a long time that gave really so much of the early epidemiology and strengthened the evidence for asymptomatic spread, emphasized the importance of testing and the need for testing, and the risk to household contacts, in that case, cabin contacts of people who were infected. Again, Keith sat in my office and said, is it possible that most of the world's cases, as we were seeing at that point, were coming from a single ship? And it wasn't, of course. Most of the cases were not from that ship, but that's what was being reported at the time or detected at the time. As Maria emphasized, countries responded to this very differently. Some really acted fast to contain it. And I think of Singapore, Australia, New Zealand, China. As Maria said, many of the countries that had experienced SARS-1 acted quickly, took it seriously, but many didn't. And some had leaders, like the leaders in Brazil and the US and the UK, who actively undermined the science and undermined their country's response to the science. At that point, I remember Dr. Tedros saying, solidarity, solidarity, solidarity. He was trying to get everybody to act in a common way, somewhat unsuccessfully. And he convened at that point a small group, an ad hoc group of experts from around the world. There were probably a dozen, maybe 2 dozen at the peak, and people would show up intermittently as they were available or when they were not being called by the leaders of their own countries to discuss the situation, share observations from their own countries, discuss issues like was masking working, border closures. That group formed the nidus for a concept that I'll come to in just a minute. But I would note that as we begin to look to the future, it's clear that there's no guarantee that the next one will be like SARS-CoV-2. And in fact, one of the things that's certain is that it won't be like SARS-CoV-2. And it could be more fatal than SARS-CoV-2. As we will hear from Colin, H5N1 is spreading much more effectively in wild bird populations and certainly in mammalian predators of wild birds, but not yet to any significant extent in humans. But the mortality of H5N1 approaching 50% reminds us that there is no guarantee that the next pandemic will be caused by a pathogen with a mortality of 1% or so, like COVID. It could certainly be double that, 2%, which would be tragic. It could be 10%, which would be, if there's a pathogen with a 10% mortality and the transmissibility of COVID, that would be a global catastrophe beyond recorded history. So what needs to happen? At the next threat, which again, no doubt will be different than SARS-CoV-2, the leading public health responders from every country need to be on the same page and need to act in unison. We need a more formalized group like the one Tedros convened, but who already know each other and trust each other's judgment, and who have practiced together on both real responses and theoretical exercises, and who have the time commitment and the authority to respond to transnational threats and determine or influence the pandemic response decisions of their countries. That's number one. Number two, because the next pandemic could also start in one small place that gets overwhelmed or spreads slowly and silently from that area for months or years, as we saw with HIV, we also need the capacity for countries to help each other, to surge respond to an area that's being overwhelmed in the case of a localized outbreak that outstrips local capacity. And this is the essence of the WHO's newly proposed Global Health Emergency Corps, which I'm really excited about and wanted to highlight. The Global Health Emergency Corps has two parts, what I just alluded to. The first part is that strengthening the interoperable surge capacity to help countries that are being overwhelmed. This builds on existing capacities that already are present in WARN, the Global Outbreak Alert and Response Network, the emergency medical teams, and a series of others already established response mechanisms. The second part is sort of what I described, a new network of country leaders from their national public health institutes or the equivalent that are able to act in unison under regional or global WHO leadership to stop the next pandemic. I think this vision is compelling, and it's beginning to gain momentum, and it's due to be launched somewhat formally at the World Health Assembly in a couple of weeks by Dr. Tedros and others. So look for that, and thank you for listening.
Thank you so much for this short presentation. There are quite many questions, but they will be put together at the end of the session if any time hopefully is left. And now from Scott, we move to the Netherlands, from Seattle to the Netherlands, and to Colin Russell, who is currently working there in Amsterdam, and he will focus on the H5N1, pandemic preparedness. So Colin, Colin's here.
Thank you very much for having me, and I'm very sorry that I can't be joining you in Portugal today. It would be nice to have a glass of wine with all of you once this talk is over. And I realize that I'm also standing between you and that glass of wine. And so with that in mind, I'll jump right in. So I was asked to speak specifically about the threat of H5N1 and about pandemic preparedness around that. And there, I think we have to think very broadly about influenza viruses in general, of which there is always a pandemic threat, but also critically consider how and what we should be thinking about H5N1 right now, particularly in light of the fact that we're all very tired of thinking about pandemics. And I think that most of the world would like to move on and talk about something else. But we are in a state where the threat is as great now as it has ever been, and we simply don't have the option to relax. So I want to start this by talking about the history of H5N1, because this isn't the first time that we've talked about it, and those of you who have paid attention to the threat from respiratory virus community for the last 20 years will feel like maybe we've just gone back in time. So in 1997, the first H5N1 viruses were detected in humans. So in early spring 1997, there was an outbreak of something in poultry flocks in rural Hong Kong. And this particular outbreak was terrifying, honestly, in large part because as a veterinary epidemiologist at the time described that this virus would basically turn the birds into liquid jello. And that's, of course, a frightening thought. But it became even more frightening when in May of 1997, a 3-year-old boy was admitted to a hospital in Hong Kong, and they simply couldn't figure out what was wrong with him. And he later died. Welina Lim, the head of virology at the Department of Health in Hong Kong, took a sample from the boy's respiratory tract and she tested it against the battery of influenza viruses that she had in her lab at that time. And much to her dismay, that virus didn't react with anything that she had in her lab. And so she sent that virus to a guy named Jan de Jong. And Jan was a technician at Erasmus Medical Center in the Netherlands. And Jan was really one of the world's foremost experts on peculiar influenza viruses. And Jan received that virus in late June, and he spent a few weeks working on it. And it didn't respond to any of the human viruses that he had in his lab. But he then tested it against a broader panel that also included avian viruses. And much to his surprise, the virus most strongly reacted against H5. So at this time, there was no evidence that the boy who had been admitted to hospital had had any interaction with pigs. And it's really important to frame this concept in light of this, because in 1997, there was a dogma in the respiratory virus community that birds simply could not give their influenza viruses directly to humans. Birds could give their viruses to pigs, and pigs could give those viruses to humans, but it wasn't possible for a bird to give that virus directly to a human. So when Jan got the positive test result for H5, he booked himself a ticket to Hong Kong the very next day. And this is literally an accurate recounting of what happened. He booked himself a flight to Hong Kong the next day because he wanted to see the lab for himself, because he wanted to rule out laboratory contamination of the specimen. Because at that point, it just didn't make sense. And if this was in fact a situation where a bird had transmitted directly to a human, the word needed to get out quickly that there was a high-risk situation at hand. Jan went to Hong Kong, and working with Wilina very closely, they ruled out any sort of laboratory contamination, and they concluded that the only plausible explanation was that a bird had directly transmitted this virus to a human, and that human had subsequently died. This is, of course, a tragedy in and of itself, but then there were about 15 other cases in Hong Kong around that same time, all of them the product of birds transmitting the virus directly to humans. Of the people who were infected, the majority of them died. Over the subsequent years, this H5N1 virus that originally was detected in Hong Kong spread across East and Southeast Asia. And here we're looking at a map of the likely route of virus spread based on evolutionary patterns inferred from virus sequence data. And so here, the two primary hypotheses about how this virus was spreading is that it was spreading via the live bird trade, and that it was also spreading via migratory birds. Now, on the one hand, sick birds typically don't fly very far, but birds also have a highly heterogeneous response in terms of the level of disease severity that they experience as a product of infection with H5N1. So it's entirely possible that migratory birds also move the virus with them as they spread across Asia. As I mentioned earlier, in the context of Hong Kong, H5N1 was a deadly infection. So up to date, there have been over 800 reported human infections, and the mortality rate has been over 50%, though that mortality rate has been highly heterogeneous across countries. Here, we're looking at countries and the number of cases observed in each of those countries over time. And here we're talking strictly about human infections. And what we see is that a huge proportion of these infections were observed in Egypt and in Indonesia. A critical component of the discussion around H5N1, though, is the observed or reported nature of infections. We have no idea of the true burden of H5N1, but given the relatively high mortality rate of human infections with this virus, it's likely that we've seen a lot of those infections. Now, interestingly, you see that that number tails off as we move to the present day, and we'll talk more about that in a moment. But all of this is to say that this was a really scary virus back in the day. Now, the other thing that's really important is that despite this huge number of cases, there was very little evidence for human-to-human transmission. There's a couple of putative cases of human-to-human transmission, but none that are virologically verifiable. Now, even still, all of this was tremendously scary, and much like in the slide Maria showed earlier in her talk, you could have been forgiven to think that H5N1 would be the next pandemic. It received a huge amount of attention. In 2012, Science had an entire issue that just had H5N1 on the cover. It was a huge threat that was perceived both publicly and in the scientific community. So in the context of the talk today, we're going to talk about what exactly H5N1 is, why H5N1 is so deadly, how you might get H5N1, And then we're going to talk about why we're talking about H5N1 now and how worried we should be as we look towards the future. So H5N1 is an influenza virus, and much like all other influenza viruses, it has a structure that looks like the schematic that we're looking at on the screen. The virus has two proteins on the cover of its on its surface. There's the hemagglutinin protein and the neuraminidase protein. Hemagglutinin is responsible for binding onto cells and pulling the virus inside, and the neuraminidase protein is effectively responsible for helping the virus break free and be able to infect other cells. Now, when we look at the line for the hemagglutinin protein, it also shows that there's a zoom-in schematic there that's labeled sialic acid. This interaction between the virus and the host cell, and it's hemagglutinin binding to sialic acid that allows the virus to infect the cell. This interaction is incredibly important, and it partially determines the host range of influenza viruses. And we'll come back to that in just a moment. But it is this combination of hemagglutinin and neuraminidase on the surface of the virus that give influenza viruses their names. So, for example, H5N1 is a combination of the fifth identified hemagglutinin and the first identified neuraminidase. And we delineate these and we number them based on whether or not they share any immunological cross-reactivity with any other known hemagglutinin or neuraminidase. So in this case, when H5 was first identified, there were only 4 hemagglutinins at that time. And when H5 was first identified, it was the 5th. Neuraminidase was the first neuraminidase in the context of N1. Now, why was H5N1 so deadly? Well, part of it was a complete lack of immunity. If we compare it to something like seasonal influenza, we have all had seasonal influenza multiple times over the course of our lives, and so we've got a fair amount of immunity built up to it. By comparison, for H5N1, we've got very little, and most of us probably have absolutely no antibody-based immunity to H5N1. This in and of itself creates a risk and makes it more like COVID than it does like seasonal flu, at least in terms of our immunity. The internal genes of the H5N1 virus at the time were also particularly interesting. Influenza viruses change their genetic segments all the time, but the H5N1 viruses that were circulating in Asia in the early 2000s, they didn't. They had a stable constellation of internal genes that they had inherited from other influenza viruses. And that same constellation of internal genes also appeared in other viruses that were cause for substantial concern and potential pandemic risk. And there I'm thinking about things like H7N9, H5N8, and other viruses. And finally, the other reason that H5N1 was so deadly is receptor binding. So here we have to go back to a discussion about sialic acid, which we were talking about a few slides ago in the context of how influenza viruses get into cells. So here we're looking at a schematic of a human lung and an avian digestive tract. Now, in birds, H5N1 is primarily a digestive tract infection, and it flows throughout the entire body. In humans, it's primarily a respiratory tract infection. Now, humans have 2 types of sialic acid in their respiratory tract. The vast majority of the respiratory tract is 2,6 sialic acid, and that includes the upper airways. The vast majority of the avian respiratory tract, by comparison, is 2,3 sialic acid. This is a small difference biochemically, but it makes a huge difference in terms of how influenza viruses see the cells that they're trying to infect. Now, interestingly, we also have the avian-like sialic acid receptors deep in our lungs, and this is part of why H5N1 is so deadly. So if we look at the schematic on the left here, labeled H1N1, the majority of seasonal influenza virus infections occur in our upper respiratory tracts. Now, this makes them easy to spread in terms of it's easy to get the virus in, easy to get the virus back out, and in general, though very miserable, they're rarely fatal. By comparison, H5N1 viruses, when they infect humans, the infection typically occurs deep in the lower lungs. Now, it's hard to get things into your lower lungs, and it's hard to get things back out of your lower lungs. But when you do have a virus infection in your lower lungs, it tends to be nasty, and that's part of why H5N1 is often fatal. It's also part of why human transmission of H5N1 viruses is rare. So, if it's hard to get things in, well, how does one get H5N1? Well, a lot of it has to do with the human-animal interface itself, and a lot of it has to do with the way that we raise poultry. So in general, most humans don't have enough exposure to wild birds to get the virus directly from wild birds. So we get the virus from birds that we have more interactions with. Here, we're looking at a poultry worker in a poultry facility in Indonesia. And this particular facility in Indonesia processes over 1 million birds per year. And you can imagine in a high density setting like this, it's very easy for a virus to spread from bird to bird. And unfortunately for the woman walking with all of those birds, well, if any of them had H5N1 and she was interacting closely with those birds, well, there's a reasonable chance that she could be exposed to the virus. She would still have to get the virus into her lungs, but it's a high-risk situation. Now, one of the statistics that I always find startling is that Indonesia is a country of 200 million people, but it has an annual chicken population of over 5 billion chickens. And when we think about how these high-density settings are distributed across a country like Indonesia or many countries around the world, you can see that the way in which we tightly aggregate livestock contributes to the threat of avian influenza viruses. And it's also people working in these sorts of high-density settings or who have very close interactions with birds who have the greatest threat of contracting H5N1. So why are we talking about H5N1 now? Well, over the last few years, the situation around H5 has changed. So in particular, H5N1 viruses, which were previously largely constrained to Egypt and East and Southeast Asia, have spread westward into Europe. And so in 2016, 2017, there was a huge expansion from East and Southeast Asia to Northern Africa and to Europe of a new lineage of H5N1 viruses. In 2021, they continued their further spread into Western Europe and back into East and Southeast Asia. And in 2021, they basically exploded all over Europe and into Western Africa and spread into the Americas. One of the ways that this is possible is because of migratory birds. Migratory birds fly all over the world, and this image never fails to sort of astonish me in terms of just how far birds can fly. But you can imagine that a virus starting off in East and Southeast Asia could easily spread around the world just by the overlap of flyways and birds spreading the virus to one another. And that's effectively the situation that we find ourselves in now. So here we're looking at the places in which H5N1 viruses have been found in the past year. And so the majority of these red dots indicate places where they have been found in birds or the environment. So this heterogeneous samples. But the long story short here is that H5N1 has now spread far beyond anywhere that we have seen it spread historically. And in a moment where you see unprecedented spread of a highly pathogenic avian influenza virus, you naturally get concerned that this is potentially a high-risk situation. And so that's why we're talking about H5N1 now. Now, the other reason we're talking about H5N1 is that the virus is now being found in lots of mammalian species, which we don't normally find H5N1 viruses in. Now, the slide I'm showing you now is one that I actually got from Ron Fouché about 15 years ago, looking at how the then H5N1 virus had spread into different animal species. Now, the geographic extent of that spread is now far wider now than it was then. However, the extent to which the other H5N1 virus was able to spread to other animal species was also impressive. And so in that way, again, whenever we see a new influenza virus spreading to a variety of new animal species, it's a cause for concern. But the situation itself is not unprecedented. So this really takes us to the question of how worried should we be? Well, the current H5N1 virus doesn't seem to be very good at infecting people, despite the fact that it is more broadly geographically distributed than it has ever been previously. So here we're looking at the number of confirmed avian influenza cases with H5N1 and deaths from 2003 up to the present day. And the key columns are on the right, where we see 2020, 2021, 22, and 23. And what we see there is that there's very few human infections, and there's also very few human deaths. Now, there's a whole debate to be had around, well, how many cases and how many deaths does it take to start a new pandemic? And we don't have a good answer for that. But the situation is substantially different than the one that we saw 15 or 20 years ago. In the context of how worried we should be, there's also factors around the virus, vaccines, and more that really shape the landscape of how concerned we should be. So first, this is a different virus than the one we were seeing 20 years ago. The H5 part is the same, but the internal genes are different. This virus has changed substantially as it's migrated across to Eastern Europe and across to the Americas. such that it's really a very different virus. It's still highly pathogenic, but it doesn't seem as capable of infecting humans as the previous H5 did. Now, how effective does a virus need to be? We don't know. And should we be concerned when we see a virus expanding its range? Absolutely. But this is a different H5 virus. Now, one of the things that is somewhat of a relief is that we do have extensive global experience making influenza vaccines. And the COVID pandemic has increased the possibility for making vaccines very rapidly. This is good news, but we need to pay very close attention to the current evolution of these H5 viruses and decide when and where we should be making new seed strains for vaccines, and at what point we should consider scaling up to make sure that we have stockpiled vaccines that are close antigenic matches to the viruses that are currently circulating in wildlife populations. There's also good news that comes from the pandemic in that we have a public that is seemingly willing to accept NPIs and ready to test. Now, if you had asked me 20 years ago if people were going to be willing to quarantine at home if they were infected with a virus like COVID, I would not have suggested that they would be. And the idea and the fact that people were so willing to accommodate lockdowns was honestly shocking to me. But it's good news in terms of our capacity to manage potential respiratory virus outbreaks in the future. However, there are some negative points, which is that we are at a point now where we are suffering from extensive global pandemic fatigue. I look at my own colleagues working in infectious diseases, and none of them would be very happy about putting on a face mask again. Unfortunately, influenza viruses don't care if we've had enough time to recover from COVID or not. As I mentioned earlier in the talk, these viruses have always posed a threat, and they continue to pose a threat. And at least to a certain extent, H5N1 potentially poses the biggest threat that it has posed so far. And so in this way, the fact that we're all very tired of thinking about pandemics is not good for our preparedness in general. The other is that H5N1 is just one of the many influenza viruses that could cause a pandemic. When we had the 2009 pandemic of H1N1 viruses, the main virus that the vast majority of the world, i.e. Scientists and public health officials were concerned about for causing a pandemic was H5N1. And we were mainly focused on East and Southeast Asia, and we were mainly focused on birds. And yet we got a pandemic of H1N1 from pigs in Mexico. So in that way, it's almost as if we were putting our attention in the wrong place. And we need to be worried about the same thing here, which is, on the one hand, H5N1 is concerning. At the same time, there are many other influenza viruses out there, many that we probably haven't even identified yet, that probably pose an equally substantial pandemic risk, but we don't know how to quantify that yet. And so in that way, we have to be vigilant in general about maintaining our surveillance capacity and our response capacity to influenza viruses in general. But this takes me to my last point, my last major point in the context of pandemic preparedness. And that is inequity in our capacity to surveil and to respond to pandemics. So here I'm going to focus just on genomic sequencing of respiratory viruses. But this extends to surveillance activities in general, and this includes diagnostic testing. And it also extends to our capacity to respond when there is a signal of concern. So in the last 2 years, more than 50% of the world's genomic sequencing data of respiratory viruses has come from approximately 5% of the world's population. Now, there's a question about how much we should sequence and how broadly we should be doing it, but we're in a situation now that is grossly inequitable. As a concrete example, Canada, which has a population of 38 million people, sequences more than 870 times as many viruses as Ethiopia, which has a population of 120 million people. Now, this is not a condemnation of Ethiopia, nor is it a condemnation of Canada. This is merely pointing out the fact that the world's resources for pathogen surveillance are wildly unequally distributed. And that poses a fundamental risk to us as a planet, because we don't know if the next pandemic virus is going to emerge in Canada or in Ethiopia or in any other country for that matter. But the fact that one country is sequencing more than 1,000 times as much as another, that's disturbing. And for those of you sitting in the audience who say, well, how representative are those statistics? You could have just cherry picked that to make an example. And you're absolutely right, I could have. But the sad part is that I didn't. This is just a random high-income country compared against a random low-income country. And in that way, the situation that we confront is dangerous, because if this amount of sequencing translates into detection capacity, Canada is very capable of detecting a new pandemic agent close to the moment that it arises, whereas Ethiopia, by comparison, is not. And the problem there is that because we don't know where a virus is going to emerge, and our only hope for stopping a pandemic early on is going to be containment at the source, we need to seriously consider how much sequencing and how much surveillance in general we should be doing where, and how that should be globally distributed. Because given the current situation involving global inequity of resources for both surveillance and response, our capacity to stop the next pandemic early is close to zero. And on that, I'll conclude, and I thank you very much for your time and listening.
Thank you so much, Colin, for this presentation. I think there are several questions now, and please.
Yeah, thanks so much, and thanks especially for those of you who have been sending questions in. We have about 8 minutes for questions. I can acknowledge for others of you who will be moderators up here, it's impossible to see the microphone, so I have no idea whether there's anybody standing at a microphone, but luckily we have some good questions that have come in online. So I'm going to start with Maria, and I have a question for you that is really about collaboration between public and private sectors. And this is a question from Sandra Chavez. So even in the space of surveillance, but also in other areas, her question is, she's surprised that it hasn't really been fully acknowledged that this collaboration between public and private, thanks so much, has been a really major lesson learned. And how do you think WHO moving forward should be engaging the private sector to continue improving those collaborations beyond the pharmaceuticals, beyond vaccine production, but engaging all players in supporting countries to establish sustainable surveillance and lab capabilities? So a bit of a long question, but really about private-public cooperation, collaboration.
Thanks, Kate, and thanks for the question. There was no real reason why I didn't highlight this one as a lessons learned. I mean, I probably could have covered many, many more different lessons. I think the fact is that the public-private partnerships that we have seen and expand across all of the different elements of the response is an asset. Whether we're talking about surveillance and we're talking about lab capacities and public health labs, veterinary labs, environmental labs. We've seen a massive expansion of looking at wastewater, looking at the different private labs that really stopped work on everything else and did work for COVID-19. I think maybe too much in one direction because we're now trying to course correct going back. We've seen the public-private partnerships across vaccination, of course. We've seen it with therapeutics. I think it's an asset that we need to leverage. Because everybody's in this space. But one of the things we know works well is the trust and collaboration that begins before these outbreaks actually happen and are nurtured over time. And I think for us at the World Health Organization, we have a lens, a global lens, and we have a public health point of view. And when we look at these types of pathogens that are all zoonotic, we need to make sure when we work across the sectors that we don't disadvantage the animal sector, we don't impact food security. We need to look at animals, plants, environments, and humans and not, put anyone at a disadvantage. But I think the public-private partnerships are ones that need to be nurtured. We need to harness the energy, the expertise towards a goal and make sure that that goal is equitable and accessible to everyone and it doesn't just benefit high-income countries.
Thanks so much. I'm going to come to our participant at the back of the room. I can't see who you are, but I can tell you're male, I think.
Yeah, I'm Koen van Driessen, pediatric infectious diseases specialist in Antwerp. So I understand the difficulties in the beginning of the pandemic with the information overload, not only all the papers that came out, but also your email boxes being swamped. And I do understand that at such a moment you have to rely on the experts. And then so my disclaimer is I'm an aerosol scientist too. And then in early April, you had this meeting with some aerosol scientists and Lydia Moravska, in my opinion, being the most knowledgeable one. and she had a very strong argument for airborne transmission of COVID being the super spreading events, because how a virus spreads most effectively, you can get that idea from the super spreading events. So I believe she made a very strong case for airborne transmission of COVID. Yet it took 2 years before it was being recognized. So if we want to, so I think something went wrong, and if we want to prevent that for the future, I think we should know what. Can you maybe tell something about why that happened that way, and how we can prevent that for the future?
Yeah, how much time you have here? I mean, one of the things that I think exactly what you point out, one of the things as we were learning, there was so little information that was coming on. And one of the things that we tried to do very early on in the pandemic was expand our partnerships to include more technical disciplines. Lydia is one of them, but many other aerosol scientists, aerobiologists, engineers to come together to expand our recommendations and our guidance on ventilation. And we acknowledged that, and we brought these groups in. We set up working groups. We not only handled it from a technical point of view, and of course there's language and terminology that is being changed currently because of this dichotomy between the droplets and the aerosols, and now infectious particles of various sizes and whatnot. And so I think it is important that you listen, that you engage, that you discuss and debate, sometimes quite fiercely. I draw the line at abuse. And I will point that out, because over the course of this pandemic, it has been incredible. But I have learned so much from aerobiologists, from engineers, and bringing them into the tent and working with. And we worked really hard to expand those partnerships. I think one of the things we will learn going forward in terms of using a precautionary principle and how we apply that as we go forward. But you do have to remember, in the beginning of this pandemic, and I think we could do a better job of communicating this. In the beginning of the pandemic, we were really struggling with masks. We recommended masks for people who were sick, people who were caring for patients from January. We did expand this because as we were learning of the choir practice and the bus, that the environment, the setting was so critically important. The role of the super spreading events and the context in which that was happening, the conversational distance versus the beyond the conversational distance, aerosols, droplets, these particles. And I think you have to be humble to continue to learn on that. And it is a massive lesson for all of us to not, anyone that spoke on TV that said that they knew the answer, we knew that they just didn't. And so for us, I think in all of the networks that you have, in the careers that you have, the advice would be to reach out, to engage, to work across disciplines, to be open, to be humble, and to acknowledge that the science changes and therefore guidance needs to change.
Yeah, that was has been and continues to be a complex area. There's a question here for Colin, and yes, let me find it again. So this is about zoonoses coming from animals consumed by humans. The question is, are one of the major responsibilities of past and future pandemic diseases related to consumption of animals by human? Is there a plan concerning animal industry and what to do about it? So Colin, is that something that you would like to address?
Sure. This is always a difficult thing because in many places in the world, we suffer from issues surrounding food access and food shortages. And in other places in the world, we're very fortunate and we have tons of food. But in this way, the industrial approach to animal rearing is very important because it provides food for many people. And yet, it also creates environments whereby infectious diseases, particularly respiratory viruses, are able to spread very quickly. And so in that way, we have to strike an important balance between biosafety and biosecurity and food security. In different parts of the world, there are different plans in place, either via governments or via the industries themselves, in terms of creating safety barriers whereby they try to reduce the spread of viruses amongst their populations and reduce the spread of those viruses to their workers. But unfortunately, this is not internationally standardized, and it's probably not possible to do in the short term. We also have a lot of people who raise animals themselves in backyards. This is also an important source of food, but it's also an environment where people work closely and live closely with their animals. This does necessarily create risks in the context of virus transmission. So in this way, I don't think that there is a good way out of it. Different countries are approaching it in different ways, but it is simply a threat that we confront.
Thanks so much.
Any
I
more
think we're
questions?
at time
There
now.
are many, many questions, and they are very hot questions, but still there is no time. So I think we should wrap up things, and thank you for your attendance. And after 10 minutes, we should begin here for our next symposium. And do not forget, what we heard today is not about the past, but it's about the future, yeah? About our next pandemic. Thank you very much indeed. Thank you.